lobster, yeah.
Squat
crab.
lobster.
It's in, it's a munid, or a munidopsis.
Check the whole rim for whatever we can find. Oopsies. All right, I'm getting shaky, sorry. Wyatt,
Yep.
thanks. All right, I'll paint it with the lasers for you again. Here we go. That's not small.
I don't see the, oh, there they are.
Jeez.
Whoa.
Yeah.
That's such a cool, like, texture.
Now that's going to be over a meter across.
Yeah, a meter and a
I think.
half,
Oh,
maybe?
Yeah,
more
meter and a
than
half.
that.
Wow.
Yeah.
Wow.
Incredible.
You can make like a sci-fi movie with that as the landscape.
Yeah, oh, wow.
There they are. Boy, I couldn't find my brittle stars there for a while. Those brittle stars, you know, that had sort of little spines on their arms, so that probably puts it in the Ophiocanthidae, and that name actually refers to the spines.
Huh.
So, Chris, let's get some clarification on what we're actually doing here.
Sure. Yeah, we got a little distracted.
Yeah, that's all good. This is important. So I'm at 2417 right now, 2417.
Okay.
Should I still keep going up slope, or what's the story?
We want to actually run along the contour here. What we want to do is, there's a sharp wall to starboard.
Okay.
And we want to see if we can run along the edge of that wall, if you can find it. So it may mean going up slope a little bit, but I can't really tell. We could be a little bit on the summit. Well, let's see. If we zoom out, you see how sharp the wall gets just due east of the summit?
Yes.
That's kind of our target, kind of like yesterday where you were working that steep wall.
Okay, you're looking for the wall and flat interface, kind of the edge of it, the ledge?
Exactly.
Understood. Okay, well, we'll head that way then.
Great, thank you.
I'm good for a ship move whenever you are.
Okay.
In the meantime, I'll look at this dead bamboo coral.
Wow, that's a pretty long one too.
Are you okay for a slightly bigger move, like
Yep.
5-0 good?
Sure.
Okay. Bridge nav? Can I get a 50 meter step due north, please? Thank you.
Oh my golly.
So that coral, how old do you think it is? Do we have any way of estimating its age?
No, but it's probably pretty old. It's probably over 100 years, I would imagine. But I don't know, given its length, that is.
So what dictates when it forms a node versus just a white part?
That's another $1 million question.
Yeah?
Yeah, I'm not really sure at all. I don't remember ever getting a good answer to that question.
And you can really see the nodes on that one.
Yeah.
Now that it's down, the tissues off.
I got a minute here.
I'll throw out something that I think is probably wrong, but I'll throw it out anyway. Those nodes could represent sort of reproduction events where they channel energy away from skeletal and they move it more into producing eggs and
down on
things
delta
like
bit,
that.
please.
But
Okay.
I don't know.
Cool. Seems like a
And
valid
that's probably
theory.
wrong.
Like a likely.
They're very evenly spaced that almost you could imagine sort of a seasonal phenomenon.
Zoom in,
Yeah.
please.
Yeah, they are relatively.
Aaron, please zoom in. Thank you.
Yeah, so you can actually see the nodes very well because this is dead. A lot of times it's got tissue covering it. So
And
those little brown patches along the stem are the nodes, and you can see how regular the internodes, the white calcified areas are. They're almost the same length all the way along here.
There's a star. There's the death culprit.
Yeah.
Murderer. Caught red-legged. There's a little,
Yeah.
interesting, right by the base there's a little lump.
Yeah, that's a node too, but it's been overgrown or over calcified.
Oh, cool.
They sometimes do that.
Okay, come wide, please.
lot of these Brazingid sea stars.
What was our bearing on that move?
It was 0, 0, 0. On the next one, I'd like to do 0, 1, 0.
A little Crystal Gorge day in there.
Or a
Small one.
black coral. Could
Or
be.
Bathypathes.
Yeah, Bathypathes. There's another one. It could be a Staropathes to the left of it. There's even a sponge up there, too, that we could look at after we look at the black corals.
Was the Bathypathy and the Styropathy both black corals?
Yes.
Cool.
Pretty much anything that's called ends with pathes is going to be a black coral.
Oh, zoom in, please.
Which also brings up another story, if you're not too burnt out by my stories already.
No, do it.
Never.
So why is this family called Antipathidae? Yeah, it's Antipathidae. These are Antipatharians.
Internal, some kind of internal brooding, maybe?
No, what does the root pathos or pathy refer to? It's
Pathological.
yeah. And what is pathological? What's that referring to? It's a problem.
Outside of normal.
It's a sickness
Yeah.
outside of normal. And antipathy means that it's
Normal.
like a cure,
Yeah.
right? And I think the original belief was that these corals had some medicinal properties. They could actually kill pathogens.
Oh,
And so they named them
Okay, come wide, please.
Antipathidae.
interesting.
So that was Staropathy, sorry, and the other one was a
What's
Bathypathy.
that little white thing to the right of
I know,
coral?
that's a sponge, and
The-
I'm hoping we're not going to ignore
on
that.
the very right?
Are you talking Argus?
No,
here,
no, no,
or-
down lower. Sorry, it just
Oh.
went out of screen on
Oh.
on the right. It's kind of parallel with the. There it is, the little white dot.
Oh, I see. Yeah, we can
Oh, yeah,
go
let's
take
check
a
that
look
out.
at that. This better be something good, Aaron.
I know, or else I'm in trouble. I'm fired. Long
In
sleeve.
trouble.
The premier videographer is looking at white dots. So, okay. Here we go. I don't know. Maybe, but
Might just be a white spot on the rock.
je
Maybe
suis
we'll find out.
skeptique.
Okay, zoom, please.
Oh,
Snail?
she's got a snail. Look at that. You did have
Nice.
something.
Nice.
Yeah, we haven't really seen any snails.
No.
I don't think this is the acrobatic snail.
Acrobatic
Doesn't
snail.
look like it.
There's a type of snail that likes to do somersaults,
Oh, that's cool.
Oh.
but it's not this guy.
This is a disappointing snail.
It's a pathological snail.
So I will go to gastropod on this one.
Yes. That's safe.
Yeah, it's very safe.
All right, come wide,
20,000
please.
foot
Yeah.
view.
Thank you. And now as there's no hermit crab, that is a living mollusk inside.
Uh-huh.
Wonder if we could take a look at that white sponge. If
This little guy here?
Yeah, this one sort of center.
Yeah.
Yep. Pretty
Yeah,
sure this is going to be another ferronematid.
Okay, zoom in, please.
Yeah. You can't really see very well how it's attached to the substrate, but sometimes their grappling hooks are pretty small.
Is this one of the Velcro sponges?
Yeah, and
Oops, bonk. Okay, come wide, please.
Yep.
I
gonna
think
move
we've
along now.
collected that already. We collected a lot of these ferronematids in the past. And we know them by a letter. That's something like
There's another
Oh,
sea
probably haven't
cucumber
gotten
we
letter
just went over.
C or D or something. I've forgotten which one it is. That's a lot of sediment for a fairly steep slope.
Mm-hmm. Wonder what's going
Yeah,
to
it's
look
good
like on top.
way to see the angle there.
Yeah, I guess, well, semi-steep, I guess.
Oh, yeah, this, yeah. It's great. Sure.
All right, creatures, where are you hiding, creatures?
Yeah, we
We
need a
wanna
little
see.
canyon. We need to protect protected canyon.
Yeah, it's a nice wall here. Come on,
It
guys.
is a nice wall, but that may be because it's just too swept by current.
I don't know if it's too swept if you've got sort of that dusting of sediment on it, though. It's the
I was 020 on that move. Oh, okay. Oh, yeah, right, a 50 meter. Yeah, understood.
Yeah, we're not quite over to that really steep lip, so I guess this isn't surprising. We're going to work our way over to the really steep part, and I think things will pick up when we get over there.
There's something ahead.
So, sea stars.
And an anemone down below it, one of the
And
same types of anemones that we already imaged. Guy again is feeding. That's why he has his arms out. And when he decides to move, he puts his arms down and he starts crawling like every other sea star.
Cool. How far do they go in a lifetime?
I don't really know, and it took a while to figure that out. I thought there was two different types, one that holds its arms up and one that is down flat on the surface, until Dr. Chris Maw at the Smithsonian Institute educated me.
About what?
About the fact that they're the same thing. That's how they move around. How can they move if they got their arms up in the water, right?
Yeah.
Yeah,
True.
and so in order to move, they got to put their arms down. In order for us to move, we actually have to take our legs off the bed and put them on the floor.
Good point. Unless you get a rolling bed.
That's true.
I wish this one looked like this one. I don't know how to do that. Is it a color thing? Everyone's doing swell, yeah.
Got a bamboo or black coral off to
Could
the
we
right.
collect a rock here for microbial analysis, or are these
This is not, I don't think this is going to be a good rock for Beth. Also, it's very consolidated. It's all glued together.
What's the, the palette thing? What's the different palettes? Oh, they're the same? Rats.
Got another one of these unbranched bamboos, and I could swear a moment ago that I saw one of those Ramuligorgias militaris that was off to the left or down there. Hard to see from a distance. Now we're slowly starting to get steeper here.
Yeah, we're kind of transiting along the slope.
I can't say I'm overly impressed with this little seamount.
All right, I'm good for another move.
It's very smooth, that's for sure.
Yeah.
So far.
There's something down there. I'm not sure what it is. This pink guy.
Venus flytrap?
I think it's an anemone. I think it's an Actinoscyphia, is a type of anemone. And why do I think that? Because it's clinging onto a dead sponge, and that particular type of anemone likes to grab onto things. It doesn't like to sit on the bottom. Venus flytrap anemone.
Okay, zoom in, please.
Yep. They're really gorgeous. I love seeing
Yeah, they
these
are
things.
beautiful. So would that pump water down its long axis?
No, it doesn't really have a long axis. It's really a gigantic mouth, and that's it.
Ah.
And it kind of wraps around there. You want to stick one of your manipulator fingers in there and see if you can get it to bite?
I mean, if you're serious, yes.
No, I'm not serious.
Okay.
Just sort of making a funny way to say that's how it feeds. Some poor animal will kind of swim in there and it'll detect it and it'll close right up and that's that.
It must be so patient.
Yeah.
You'd have to be.
All right. Thank you.
Eat like once every month. That.
But can you visualize it? We're just laying on our bed and we just keep our mouth open and wait for the food to kind of drop in. It's not that on a
I
truck.
can
Roman
visualize
Emperor.
Who would you want to drop food in for you? Your bed must have to be under a tree,
Yes,
or
that's true. Yeah, you got to be careful what actually falls in your...
I mean, even sleeping underneath an apple tree and you wait with your mouth open. Imagine
Oh,
an apple.
sorry, excuse me. Crinoid,
Oh, stock
crinoid,
crinoid. Yeah, I see it.
crinoid. Sorry to interrupt, but I've been looking for a stock crinoid the whole
Clearly
time.
more important.
I've been waiting to see one of these guys. I think that's it. I'm not positive, but...
It's doctor isn't on something dead.
Yeah, we're not sampling this, are we?
Let's take a look at it first.
Okay. Okay, zoom in.
Oh, no, it is stocked.
Oh.
Yeah, it is. And this is a... God, I'm so rusty on these things. Having so much... Oh, here we go. All right. Is that what... Yeah, it could be. So I think this is what's called a bathycrinus. I'm not positive. Wait a second. Let me look at a few others here.
If you'd like to sample this, I'd like to know sooner than later, please.
No, I don't think we want to sample that. It's not different enough.
Okay.
Yeah, so I think it's bathycrinus, possibly. I gotta look right underneath those arms. It doesn't really have much of a disc under there. And those are hydroids that are growing all over the stalk.
Oh, is that what that is?
So yeah, I'd go bathycrinidae at least, and possibly bathycrinus is the genus. The reason I love these things is because they look almost completely unchanged from the crinoids that we see in the fossil record. No, it's not a coral. It's an echinoderm. And so is this one, but this is a type of sea star that we're on right now.
Okay, thank you.
Yeah, thank you very much for that. Sorry for the interruption. But
No,
I had to get my
Sorry.
I had to get my stock
do
crinoid.
not worry about that.
Oh, you've been jonesing for a stock crinoid.
Can I use Bubble for a gauge check really quick?
Absolutely.
Okay.
This is too much sediment. Corals don't like this much sediment.
Oh, that's a lot, too.
Yeah, I'm surprised.
How far do we go until we get to that steeper contour
it's,
zone?
well, I dropped a marker. It's 400 meters away, but let's see where that is.
Oh, yeah.
Up there, yeah, it's pretty far.
Pretty far. Okay.
Oh, we still got a ways before we get to
Till we
really
get to the steepest. I mean, this is still fairly steep, but if I'm understanding you correctly, if you're looking at high pack, this looks like the way steep area. Yeah.
You're right. Okay, well, that's good. I'm glad because we're heading, well, hopefully we'll see more stuff up ahead.
Bridge Nav, I think your mic's open.
A viewer wants to know if sea stars swim. If there's a species of sea star that swims.
That's actually, boy, that's a good question. No, not that I know of, but there are species of crinoids, the ones that don't have the long stalk, that are called feather stars, actually, that do swim.
Mm.
So that's a good question. I had to think about that for a bit because I don't know of any that do. You know, you viewers out there, if you're not too shy, you could put your first name in there and with a question, because we're getting some good questions here, and we'll certainly address it directly to you if you give us your first name. Address our answers to you.
Doesn't even have to be real.
Yeah, we got a lot of the viewers identified themselves when we started our watch. So
Oh,
that
is that
really
right?
neat.
Oh,
Yeah.
I haven't been looking over your shoulder because we had to go down and get tested for COVID and all that stuff.
Yeah, we got lots. Ricky from Adelaide, Australia, too.
Oh, it's Ricky. Okay, great. Okay, we'll keep a better eye
So
on
quite a lot
this.
to go. 20 meters.
Sure.
You want to go faster? 100 meter step?
Sure.
Oh, wow. Okay. Bridge nav.
Reese from Canada again, I think.
Can we get
Another
a
Canadian, yeah.
She or he was
100
here
meter step
last
bearing
night.
020?
Calgary, Alberta, Canada.
Crazy, I know.
Oh, Reese, okay. Yeah, she was there last night.
Yeah.
Multiple Canadians. Whoop, that's the end of my leash. Just gonna hang out here and look at this dirt for a while.
Yeah, wow.
A viewer is asking if echinoderms and echidnas are named for the same from the same root word.
Same root word?
Mm-hmm.
Yeah, the Latin word. You know what? I've got a Latin root thing, and I'm going to go. So an echidna, there's a type of
Echidna.
a species of a moray eel that has that, and kind of, I think it means another word for spiny, but actually I don't know. Let me see. My little handy Latin
I'm very
translator.
impressed, Chris.
Echine, a hedgehog, a sea urchin. Well, that's helpful.
Spiky
Well, they're
back.
both spiny. Yeah.
And then it was echidnas is what he was saying. So
I was going to say there is a mammal that is.
A mammal's
echidna.
echidna, yeah.
okay. ECHI is referring to a viper or an adder, and ECHIN is spiny sea urchin type of thing. So no, so the answer is no, they don't have the same word roots. And who is it? Is that from
No name for that one.
Oh, okay.
We have another question from Melissa
Okay.
from Ohio. Are sea stars of the deep also being affected by wasting syndrome?
I don't know what wasting syndrome
Oh, the
is.
mass
I'm afraid.
wasting
I'm sorry,
syndrome.
Melissa. Can you tell me what wasting syndrome is?
No,
It
as
sounds-
far as I'm aware, no. It's
What is
it's
that?
on the west coast. It's where mass die-offs of sea stars along the west coast of the U.S., but I haven't heard of anything similar in the deep ocean species.
That was a great question.
Yeah, it sure is. And we've got Moya, who's from New Zealand originally, who's joining from
For 8 years.
yeah,
8 years.
for 8 years.
I was going to say Moya. Moya.
Moira?
Yeah.
Yeah, she's
Moya?
on right now.
Yeah,
M-O-Y-A.
long
Hi.
time fan.
She's been a long time fan.
Okay, this is another question, and it's part of our objectives too on these dives. How do you decide which live samples to take and which ones to leave?
And this is from Reese. And most of the decision, we have some requests from specific taxonomists to try to collect a certain type of animal because they're trying to figure out that animal. There may have been a collection of it before, but they don't have enough information. So it's that type of request. And we do have a couple of those on this cruise that have notified us to be on the lookout for a few things. But aside from that, we're trying to find things that are new and unusual. And we actually have what we call a hit list or a wish list. And it's animals that we haven't generally been able to identify even to genus.
Kind
And
of dig that one.
we know they exist. We have pictures of them. but they haven't been collected and we're not sure. And so those are the ones that we want to collect because those are potential new species. And then the third criteria is if we come into a big community and there's a whole lot of one type of species and we're not sure of what it is, we'll take a piece of that because we realize it's a very important member of the community and we really need to know what that species is, even if perhaps we suspect what it might be. So those are kind of the three criteria that we use to decide what to collect. I wonder if we've got some possible rocks here.
Oh, there's quite a few rocks
thoroughly
to choose
enjoying
from.
the substrate.
Well, we can have a gander.
Yeah.
Can you come down in delta, please?
Yes.
At about 13, maybe?
1-3.
Yeah.
I don't see the lasers on again unless they're there. Aha. Okay. I don't know. Help me figure out a size there, pilot. We're looking for angular. You already know that.
This is the Andrea rock?
Yeah, an Andrea rock. I was thinking I would let Beth collect her own there because she knows specifically what she wants.
Sure.
Since she only has 2 boxes.
Well, we'll just keep side swiping as we are on the go here and keep our eyes peeled.
What about that one? Coming up the middle center,
Lower.
has a little shadow on it.
I mean, sure, 20 centimeters wide. 25.
Let's take a look at that. That's a possibility.
Just looks like it's been sheared off, so it's kind of more angular.
Do you want me to hold?
There might be a critter on the left side
Holding?
of it too.
Oh.
And we have a follow-up question to the previous
Really?
one.
Oh, all right.
Is there a risk of removing a unique specimen and impacting a reproductive population pool? Or are most species being collected numerous enough that this is not a concern?
I'm going to answer that in a second. I think that's a keeper. So let's go ahead and put that in the big box with the other one. Generally speaking, we might see only one because we're only sampling an extremely small part of the seamount, and there could be thousands of a particular thing, but we only come across one. And so we're not too concerned about that. You know,
Trevor wants
generally
to know if he
where
can
there's
put
one.
this in echo. You're not on SPL.
Oh.
He wants to know if he can put this in echo.
Oh yeah, echo is open.
Thank you.
And then yeah,
Sorry.
that's where the other one is.
Okay.
Sorry.
No, that's my fault. Completely my
No,
fault.
I was looking over at the chat at the time.
Oh, that wasn't even a speed record. Come on.
That was pretty impressive, though.
Anyway, chances if
All
we
right,
see
dice
one,
level,
there's
please.
some more around, and so we're pretty confident
Jeez,
that
oh,
we're not going
man,
to really
Trevor.
affect the population for that question.
Bonk.
They can be very spaced out, and also these animals in the deep sea are often on multiple seamounts, even in very low densities. They're scattered all around. That's one of the reasons
Bridge nav, another step, 100 meters, 020, keep speed, thanks.
you will very rarely hear us use the word endemic, which implies that it's a new species that's associated just with a specific site.
Oh, there's some sponges right there.
Oh, yeah, there is.
Oh.
I got to play catch up here.
Okay.
We'll stop with the next ones, though, I promise.
Well, the next one's right there on the left, but no.
The next ones.
Just kidding.
Okay.
I mean, sure, what the heck. What could go wrong?
We don't have to stop, honestly. I was just,
I know. I'm actually ahead again, so.
there's one
Put it in
straight
boost
down.
mode. Just there's one
Right,
to the
right.
left of the lasers
Right
that you're looking for?
there.
Yeah.
Yep.
Another
Okay, go
rock.
ahead
Oh,
and zoom,
sponge.
please. Spongy.
We have seen this one before.
Yeah, so this is in the genus Semperella, and boy, I'm tempted on these. We don't really know this genus very well. Cha-cha-cha.
The pressure's on, Chris.
Can we slide around a little bit and look at the right side of it? Is that possible, or is that kind of hard to do?
I can do that. The longer we wait, the less time we have to sample.
Or we could just stop the ship.
Well, we're gonna have to go back to sample, so I have to be quick no matter what.
Let's wait on this. I saw
Okay,
a
come
few of
wide,
them,
please.
and we might like to.
We are in, just so you know, Chris, we're in kind of speed mode.
Yeah, that's fine. Okay, to get
so-
to the ledge.
Yeah, we're trying to
samples
get
have to
steep.
either be fast or really, really slow because we have to bring the ship back.
I got it. Okay.
Yeah, Argus and Herc are about 20 meters off the bow. If that gives you any context.
Okay.
Which again, both of those options are totally fine. We can do the sampling, but so you're aware of the operational constraints.
Oh, there's a lot of sediment here.
Yeah, this is not good. Well, it's good if we might see some different things, and we might see some more fish for Ken.
Yeah.
Good, yeah, fish, I was wondering.
Definitely see things disturbing, digging up the sediment. You see those kind of lighter areas. It's typically some kind of sediment-dwelling organism.
So,
Yeah, that's right. They're probably underneath the sediment, kind of digging burrows and things. And that's why a lot of fish like to hang out in sediment, because they kind of know there are things in the sediment. There's, what is that? I don't know what that was. Never mind.
so Zoe, a Hawaiian in Australia, wants to know if you have ever discovered a new species on a Nautilus expedition.
Oh, yeah, absolutely.
Yeah, that's
We got some new species of squat lobsters. I'm pretty sure we got some new species of sea cucumbers in 2018, but the problem is that taxonomy takes quite a few years. A lot of research that goes into describing
What's
these.
that little thing at the bottom of the screen near the middle?
Oh, that's a sea cuc. I don't know if that's possible.
Looks like
Cucumbers, speaking of which.
Zoom in on the cucumbers, please.
a very nice one.
That's an interesting one.
So you can't really stay here for a second while I try to very quickly look this up.
Sure, yeah.
Okay. This is a potential collectible. I wonder if you could slurp it.
You can stop the ship.
Okay.
It's going to be
Bridge
really
nav, hold
squishy to
position.
grab. That's all.
Okay, come wide, please.
And we actually slurped 3 cucumbers in 2018.
Come full extra super wide. I might have to come back for it, but I'll try to be quick. We stop
Okay.
the ship just in case.
Okay. So the reason this is a
Camera on
collection,
slurp jar, please.
we don't even know what family this is to answer
Right.
the question. We have a guess. We think it possibly is a late magonid, but
Yeah, that looks like it. Late Magoning.
You can head Argus, have Argus's heading be
Thanks,
020, 040, something like that.
Okay.
The slurper.
Sorry, I'm coming around the other way. 040?
Yeah.
Okay.
Oh, what? Come on. That's not fast.
Are all jars open?
I think all jars are open. We
Yep,
launched.
all jars are open.
All right, ready for
Okay.
slurping.
Okay, zoom in, please, video. And go ahead and give me, I don't know, 40%. Let's try 30% to start.
Okay, 30%.
Oh,
Boy, that's slurp
nice.
size.
Oh,
Okay,
it
come
works
wide,
pretty
please.
well for these guys. Okay, we're going to look
That
at
was
the
it.
Can you
jar.
show on porch light, please?
That was a professional slurp if I've ever seen one.
Yeah,
There he
that was
goes.
smooth.
Where is he?
He just went up on the right. So he's halfway to his
Oh,
home.
there he
is.
is.
Yes.
Wait, see, it works really well for these guys. And they come up in really good
Yeah,
shape.
you can kill slurp.
Okay.
Thank you very much.
And then bring her to flush and flush it out. Whoops, hello.
Oh,
Oops.
that's his buddy. He's like, you took my friend.
I was an anemone. That
Yeah.
was
Oh yeah, that was at the limit. That was just about to get tugged. Perfect. Yeah, flush at your leisure.
good job. Thank you, pilot. Appreciate it.
Thanks for the opportunity.
All right.
And you can bring Argus's heading around again. See if you can find me.
Okay.
Nice. That was fun.
Okay.
So those
Hello.
don't turn to complete mush when they get up to the surface, huh?
Excuse me?
They don't turn to complete mush as they bring them up to the surface.
Some are more durable than others. That guy could be a little mushy,
Have you flushed?
but he's in water and so he'll stay intact.
Oh.
And we'll transfer him very delicately. I've seen this before. We may even use spoons or scoops to get him out.
Oh.
Yeah.
You good for ship to start moving again?
Yeah.
Bridge nav, can we go back to
Did
that
you come around to
100
port?
meter step,
Oh, you came around to port, I
020,
see.
0.5 knots?
Yeah, you're good. Yeah.
Thank you.
I was wondering what you did do. Roger. Roger, roger. All right, now I got time to move this guy away.
So as it's getting steeper, we're getting more and more sediment.
Yeah, this is just one big sand slope. Or,
It's flushing.
or
Well,
snow.
Let's look for sand critters.
Mm-hmm.
Such as sea cucumbers or
All right,
sand
nice teamwork.
critters.
Okay, we have quite a few more questions here from the audience.
Okay.
Which comes first, the expedition location, then the scientists with their wish lists, or vice versa?
Great question.
Usually it's the location in this case,
Mm.
because we are dedicated to trying to help the big monuments, the really beautiful reserves and so forth. And so we were specifically looking for dive sites within this monument. And when we settle on these seamounts, then we start asking.
Don't you have something to do?
what
Sorry,
type-
Chris, that was directed to Justin, not you. We have a pest up here in the front row.
What was that?
I just got salty with Justin on SPL, and I thought maybe you thought I was talking to you.
Oh, no, I didn't.
Okay, good. Sorry. Carry on.
Yeah, that question was from Patricia in Tennessee. So thank you for that question, Patricia.
Yeah, thank you.
Zoe again, because you are having to collect samples for other researchers, how much do you need to know about the specific research and sample requirements of the other researchers, not on your watch?
These are really good questions.
Yeah.
We know a bit, let's say this much. So when a researcher contacts us, they usually would want to justify why they're making the request. So they'll provide us with some information on why they think it's important. So, but we don't know all the details. So for example, we have a request in for a certain type of bamboo coral, and we haven't seen it yet, but we're looking for it. And it's a problem bamboo that they're not sure about, and there's graduate students who are involved who are trying to do research on it. So there's some good reasons. Note, these particular sea cucumbers are also for a graduate study. And so, yeah, there are just various reasons to do it.
Is that a sea anemone at the bottom of the screen there?
Uh-huh. Probably another actinostolid.
Do species on different seamounts exhibit divergent evolution like species on island chains?
Like I said, these are good questions.
Yeah.
We get some
I honestly don't think we know enough because we just haven't been on enough seamounts yet.
Uh-huh.
You know, the estimate, depending on how you define a seamount, there are estimates between 50 to 100,000 seamounts in the Pacific alone. And everybody defines it a little differently. It's on their height and size. But we've been on maybe a few hundred, all the ships together.
Uh-huh.
And so we just don't know. We just don't have enough information yet.
Okay. And Jeff listener is got some information about the squid that we saw at the beginning of our dive. He says that he had Dr. Kat Bolstad take a look at the squid, and she confirmed it was a Tanginia, aka the Dana octopus squid.
Wow, that's great. I have no idea what that is. Well, thank you very much, Jeff.
Yeah.
That's awesome.
That is awesome. We're going to keep that record.
Yeah.
Can you put that in the record? You just
So,
put it in? Yeah, we just put that in there. Thanks very much.
yeah, so Tanginia, aka Dana octopus squid.
He's talking about that transparent one?
The one
Is
we
that
first saw at the
I
beginning
think-
of the dive.
Oh, okay, with the big fins.
Yep,
Okay.
halfway down. A lot of sediment still.
I mean, oh, joy.
Chris doesn't like sediment because the sponges don't like sediment. So Chris
We're
is part of the
hauling. We're
sponge.
trying to get through it.
Yeah, there are some sponges that like sediment.
Oh, yeah?
Yeah.
Like what?
Well, there's one that's called Hercules in Hawaii. And it likes sediment.
Okay.
There are some in a group that it's a subfamily Euplectellinae and Euplectellids, and they actually have that grappling system, but they use it in sediment.
Yeah.
There's another cucumber.
Yes, it is.
Go take a look at it. Just it looks a little different.
You can zoom in on that, please, Aaron. Is it the same one?
Nope, it's a different one.
Is that the same family, Chris? Or species?
That
That late
Are we slurping?
all gonna die?
No. Give me a second. I know you're trying to rush. Okay, let's slurp if you think you can do it.
Yep.
You had
Rich,
so much
wine,
fun.
hold
please.
position, please. Thank you.
Oh, if we even have this one in the gallery here.
It's a good thing they're not very jumpy.
I can tell you that we haven't collected this before.
All right, bubble on the arm, please. In preparation for once I've slurped it. Oh, no, I'm not doing that. Never mind. You can...
Where's it
Hey,
going?
come back.
What are you doing?
Oh.
Stay still.
Saying, I don't want to go.
You can switch the cameras around there, Antonella.
It's making a tumble for it.
So some cucumbers swim that are down here,
Oh,
but
no.
that does not constitute swimming. That's like floating in an air
No.
air
Oh,
mattress.
no.
Or
Yeah,
something.
it's like being picked up and carried away.
Silly little coral cutters. There we go.
I didn't even think of that, but I guess it is easier with the parallel jaws, right?
Absolutely. It's okay. It's just challenging.
Is it going to fit?
Okay, slurp action, please. Are we? Wait, we're on flush jar. We got to be on a different jar. Seven, sure. Seven's empty, right?
Yeah, 7 is empty.
Sure. Oh, maybe he's too big. I got to come back to it. Hold on.
It's kind of chunky.
And can you please turn Argus heading? Turn to starboard 90 degrees. Facing 045. Yeah, he's too big. It's not going to fit in the tube. You can see the tube compared to him.
Yeah.
Okay, I'm getting pulled too. So
okay,
can we bail out
that's
on
fine.
this?
Yep.
Yeah.
Boy, did you look out. sea
Yeah.
cucumber.
Too large for slurping.
Yeah.
Shucks.
Lucky day.
That's a defense strategy. They
Being too
lake
large for
themselves.
slurping.
And Bobby from Burbank wants to know if any creature has escaped the slurp.
Has escaped the slurp?
Well,
Yeah.
that one just did.
I think we just saw one.
Oh, well, by swimming back up to the tube
Oh, no.
against the suction.
No,
No.
No.
it's too strong, and once they're in the jar, I'm afraid that's it.
Ready to go?
Nope.
Nope. Okay.
Can't turn yet.
Okay. We're all action.
Yeah.
Okay, let's
All
do a
right,
little sour grapes. I
turn
didn't want that
and-
sea cucumber anyway.
Yeah, that was a
Ah.
Who
stupid one.
cares?
Yeah. Didn't look frilly enough for us.
Oh,
Okay, we
sponge?
can do a ship move now.
Sponge.
Okay.
But
Unless you want to look at that sponge.
no, I know what it is. We
Okay.
can go by. This is Hyalonema corynonema.
Bridge and nav.
And you can find me again on.
Can we get a 100-meter step, 020? Thank you.
So I asked for that heading change just so I have a little extra tether room before I get tugged.
Yeah, that makes sense.
So that's what you're just having her do just a
Face
little bit.
away, yeah.
Yeah,
Maximum reach.
got it.
This might be a stupid question, but is there any chance it might be a little better slightly upslope?
I think you would have as good an idea as we would, but happy to explore wherever you want.
You want to sort of angle a little bit upslope, and we'll just sort of
Sure.
take a peek a little bit?
How about something like that?
Yeah, go right to that target.
Yeah, is that all right? 350?
Sure.
Bridge and nav. Can we change that bearing to 350? Yep, 350. Thank you.
Yeah, I think that sea cucumber might have made it a little ways up the pipe and then just got
Yeah.
plugged. It might have gone through. I don't know.
Hard to say, you know, some of them are really flexible, a lot of water, and other ones are a little more durable. If he was a durable one, he could have gotten stuck in there.
I love that way to describe an animal, durable.
Everything in relation to the slurp.
Yeah, I asked for the wrong thing there with the bubble cam. I didn't want that at all. I just said, hey, bubble. And I'm like, nope, that's for a rock sample. That's something entirely different. That was my bad. Yeah, I
Nice
also got
pillows
there
down
eventually.
there.
I
See how they're
didn't
kind of etched by the sand?
Uh-huh.
You can see there.
So we have a question from a viewer. What did the left behind sea cucumber say to his sampled friend? What did the left behind sea cucumber say to his sampled friend?
That sucks. Oh, jeez. Oh. Uh-oh. You drew first blood. Now you've started it. Oh, my goodness.
So a more serious question.
Than that, really?
Well, that's kind of fun. It's a reasonable joke. I like it.
I like it. Yeah.
that was fun.
What data do you collect and how do you store it these days? This comes from Ed from Phoenix. In the 80s, he worked with the USGS Pacific Marine Geology and spent a little time as a science watchstander.
Okay.
So,
well, let's talk about specimens. We collect exactly where the specimen was collected. That is the latitude, the longitude, the depth. We also record temperature, oxygen concentration, salinity, pressure, and when the dives are actually annotated, the video of that specimen will get the type of substrate it's on, what the general substrate is. If the animal is on some other type of animal, if it had some commensal animal associated with it, these are all linked. And there's actually a data form that the video annotators, this is done at the University of Hawaii and other places, that we fill out. I used to do it, but now I've retired and the team is still doing it. There's a database template that has 73 fields that we have to fill out for each observation from the video. So also from the video, we do counts. We do size measurements where possible, where the lasers actually are near the animal. We have odd observations, the condition of the animal. If the coral is kind of partially dead or something, that is noted. We basically try to wring out every single drop of data we possibly can. It's just too important. These visits down to this realm are just too infrequent, so we try to take full advantage of them.
Where's it
What
stored?
is that curly thing down there?
Curly thing.
Yeah, I wonder if that's...
The rock itself?
Yeah, you went over it a little bit. It's probably just a sediment mirage or something, so I guess we keep going on. I think that's a broken pillow right there, and you're seeing part of the rim, right where the lasers went.
It's one of those old down Faraday sponges.
What would cause that break?
Just they're not, if it was sort of hollow, it's a little more fragile. And so sometimes just it breaks off. Even when it's erupting, it could break off.
What's that?
And when it does break, they're called truncated pillows.
Kind of going the wrong way here, but I see a cool thing in Argus view. So
Left, yeah.
Yep.
Oh, yeah.
Another sponge. Glass sponge.
Did he just call a sponge cool?
yeah,
Sponges
absolutely.
are cool.
Sponges are cool. Just kidding.
Oh.
Squirrel.
The bubblegum coral?
It was a nice try. It's not a bubblegum coral, I'm afraid. So Steve,
You know better to ask now.
Right, so what is it if it's not a bubblegum coral, pilot? What is it?
Another pink one? I don't know.
Oh, coralliid.
Coralliid.
Yeah, hemicorallium.
I think I've tried to avoid learning that.
Oh, can you see that right by the
What's
lasers?
that little
See that
thing?
thing that's moving?
Yep.
That's
Oh,
that's our
that's a
acrobatic
fish.
snail.
Okay, zoom in, please.
Oh, really?
Acrobatic
Wow.
snail.
Acrobatic
Oh,
snail.
no
Oh,
kidding.
Oh,
wow.
wow. That's cool.
Why is it moving so fast? And
Because
the light,
it's
or?
an escape response or something because of the lights.
Wow, fascinating. That's super
Wow.
cool.
Seems
They
like
actually
it's
call it
like,
an
a
acrobatic
good
snail?
it's like
I'm
just
gonna give you-
asking to get eaten. If you
Yeah, we have an exact name for that guy. Sorry, I'm 3 years out of date on these things, so give me a second.
You're going the way I'm not going, buddy. Come on.
that's
pretty
that's what
fast.
he knows.
Oh,
there's
oh,
another
there's
one.
another
Oh, it's a
one.
It's a circus
Family.
party. The snail
It's a relay
circus.
race. Yeah.
That's wild.
Okay, I got to move along. Come wide, please.
Okay, where's
you.
my acrobatic snails?
You weren't kidding. They really do flip.
That was acrobatic, yeah.
Bridge nav.
I thought maybe it was going to be like a little roll. Like
Can we
very
get a
slow?
50 meter step 350?
That's a lot of energy to expend
Thank you.
to go through that display.
Yeah. I wonder if they sustain
pretty
any
steep.
injuries.
Oh, for crying out loud, I can't find it. I know it's in the guide. Come on.
Oh, there's another feather.
Is this him?
Or
Nope.
sea lily.
Come on, where's the acrobatic snail? Okay. Where's the
Oh, cool
online
Argus
guide?
view right
I gotta
now.
look online.
Yeah.
I know it's on the online guide.
Super cool.
Yeah.
All right, and that's the last time I'll do that.
That was cool.
I see you.
That's very
That's
scary
cool.
to me. All right, come up on Delta, please.
That was really cool.
I don't like doing that, but it's very fun.
Got it, Megan. Thank you for the prompting.
Ah, I got it, finally.
Yay.
Excuse me. This is Gaza Daedala.
Gaza.
Yeah.
Oh.
That's the guy right there.
Okay.
He's not doing his acrobatics. He actually stops so we can get a picture of him. But that's the acrobatic
thank you.
snail.
Chris, I
And
think you're like,
there he is too. Just, they could be shinier or duller or anything like that.
I think we are in the steeps, so I don't know if you want to think about doing like transects up and down the steep slope to see if you hit what you want to see, or if you want any other change in plans.
Okay, I see where we are. I was thinking let's go up a little bit further toward the rim and just see if, I mean, this substrate is starting to look a little more promising.
Okay, yeah, we are headed up that way, so
Yeah.
no worries.
What's that little red thing?
Oh,
Don't know. I don't have a lot of time here, but I can.
Oh, there it is. I
Quick
was
gander.
trying to see
Okay, zoom
what
in,
it was.
please. Give me a shaky
Oh.
zoom, but
Yep.
It's Anthomastus or a mushroom coral.
Cool.
cool.
There you go.
Okay, thanks.
Now known as Heteropolypus. I'm getting better at saying that, aren't I?
Pleuropolypus?
Heteropolypus.
Heteropolypus.
Starting to actually
heads.
like it. There's an orange thing. What that orange thing is, I wonder if we could take a look at it. It's probably a black coral. But quick look.
One of
Line it
our
up on it.
viewers.
Okay, zoom in, please.
Yep, this is another Staropathes, thank you.
Nice.
One of the viewers, what?
Okay, thanks.
Yeah,
Come on.
said that
Oh, you okay to keep this up smaller now so we can
the
explore?
acrobatic snails were hilarious.
Bridge now.
Ah, hilarious.
Oh,
Nice
Yeah,
one.
20 meter step, 350.
uh-oh, now we're going to have an avalanche of those.
Yeah.
Squat lobster and something.
Yeah, I wonder how many viewers ever knew that snails could move like that. I sure didn't until I
did
saw
I
it.
did not
know.
know that.
So that was
Definitely
really
didn't
something.
know.
It's really something, yeah.
From Muddy Netherlands, someone is asking, do you store the video data on board the ship?
No, it's too hard. And so there's a whole laboratory back in the University of Hawaii, and there's a number of laboratories around that do this. And we use some software that was developed by the Monterey Bay Aquarium Research Institute that's called VARS, the Video Annotation and Reference System, which is amazing. And they customized it for our lab. Twice they brought their technician out and got it to work just the way we want to. And now that lab is being headed by Dr. Jeff Drazen, and there's a couple other people, Megan Putz and Sarah Bingo, and a newest member, Tiffany Martinez, I believe is her name. I've met her only on Zoom. She's a new member of the team.
What do you call that? The VARS? What was that? What did that
VARS, Video Annotation and Reference System, and to me it's absolutely amazing software.
say? What does it do?
They developed it. They're the ones kind of the pioneers on this whole video annotation thing. So the first thing you do is you have to populate a whole reference base with everything, every type of substrate, every type of thing that you want to record in your data.
Wow.
And there's another CQ, kind of the one that didn't make it.
Yeah.
But anyway, and then VARS controls the video, so you get digital versions of it. And you can stop and
I
pause
prefer
and
upslope.
reverse